Our method is one of the first to be able to dissect the genetic basis of trait differences on a genome-wide scale between species instead of within species.
The main advantages of this technique are that it is high throughput, high resolution, and widely applicable.
To begin this procedure, retrieve a JR507 freezer stock strain from storage at -80 degrees Celsius and streak it out to single colonies on a YPD agar plate. Incubate at 26 degrees Celsius for 2 days or until colonies appear. Then inoculate 100 milliliters of liquid YPD in a 250 milliliter glass flask with a single colony of JR507 and incubate at 28 degrees Celsius while shaking at 200 rpm for 24 hours or until stationary phase is reached. The next day, measure the OD600 of the overnight culture. Back-dilute some of the overnight culture with fresh liquid YPD into a new 1-liter flask to create a new culture with an OD600 of 0.2 and a volume of 500 milliliters. Repeat this process 3 more times to create a total of 4 cultures with an OD600 of 0.2, using the same overnight culture for all of the new cultures. Incubate all of the cultures at 28 degrees Celsius for 6 hours while shaking at 200 rpm. After this, combine 2 of the 500 milliliter cultures to create a 1-liter culture. Combine the remaining 2 500 milliliter cultures to create a second 1-liter culture. First, split each of the